We are the paradoxical ape. Bipedal, naked, large-brained, long the master of fire, tools, and language, but still trying to understand ourselves. Aware that death is inevitable, yet filled with optimism. We grow up slowly. We hand down knowledge. We empathize and deceive. We shape the future from our shared understanding of the past. CARTA brings together experts from diverse disciplines to exchange insights on who we are and how we got here. An exploration made possible by the generosity of humans like you.
Thank you everyone for coming, and I'll just take a moment to tell you a little bit about how we decided to make like a CARTA meeting entirely on human imagination. And this is one of those things that it seems to happen only in San Diego, where two people, and I'm talking about myself, who I'm a professor in pediatrics and cellular molecular medicine, turns out to be in a meeting sitting together with Sheldon Brown, who's going to hear as a first speaker. And Sheldon Brown comes from visual arts. So a scientist and an artist, we start talking, and we realize that we have something in common. We are both fascinated by human imagination. So I make a challenge to Sheldon and say, well, can we put this in a scientific contest? And we start planning about a meeting like that, and we realize that most of the time, when we try to understand something, if it's unique or enhanced in humans, we turn on to compare to our closest living relatives, such as the chimpanzee. And comparing to other animals is difficult, especially if you're talking about cognition and imagination. So this time, I think you're going to hear lots of speakers talking about imagination in modern humans and also in extinct humans, such as the Neanderthals. And we hope that the combination of all those speakers, all this knowledge will help us to understand a little bit about ourselves and answer those questions that Ajit posed to us.
Okay, so we're going to move into a new space in time right now. We're going to move into the night. Some 3,500 years ago, humans engineered control of fire. It's actually quite complicated. There was a huge caloric gain from food because cooked food makes many more calories available. Protection from predators. But what's most interesting about it is humans extended the day by 4 or 5 hours to become the shortest sleepers of all primates. And the time that gained was economically unproductive. No more calories were going in, nothing was happening economically. And yet the physical changes in circadian rhythms were enormous to extend the day. And there were some very major impacts on health from reduction in melatonin and other things. So there were costs. So the question is, why? Why was this selected for? Why didn't we just go to sleep? So what I'm going to suggest here is that Firelight opened a new time and space for the imagination and for the arts. There's the wonderful quote from a German writer, "Nobody sees anything in the night. Everybody sees something in the night." So the night is often the time of the imagination. If I would define the imagination for this talk as the ability to form mental images of something not present to the senses and never wholly perceived in reality. So what transpires by day and night? Why do Firelight hours matter? Well, this is not so easy to find out, but I became very interested among the Junkwasi Bushmen of Botswana and Namibia. These are a group of people who live by hunting and gathering. They utilize about 100 plant species, 30 animal species, and in the hard times of year, they even have to gather water. The only water they get is from melon or from roots. And they live in very small camps of 25 to 40 people with egalitarian structure, age hierarchy, and recognized camp leaders. So the interaction, the scale of interaction in everyday life is very, very small. And yet they have these incredibly sophisticated social institutions. And the question is, when you live on a postage stamp, how do you see the big picture? So that was my question, to see what happens in the day and the night. Here are some of the cultural institutions that they have. Complex kinship systems, defining both biological and imaginary or fictive kin. These systems are so complicated that I, after 30 years of working there, am still trying to figure them out. Rules for widespread food sharing. They have arranged marriage and elaborate marriage rites, sophisticated systems of land tenure, trans-healing in an unbelievable communication with the spirit world, and then networks of harrow exchange, widespread access to the resources of others. People move around. We'll see this. So what happens at day and night? That was my question. And with data in 1974, I collected information on day and night conversations with groups of 5 or more adults, longer than 15 to 20 minutes. And then 2011 and 2013, I collected the full stories and had them transcribed and translated by a team of Junkwasi who are now working with laptops and sending me the most amazing emails. All the gossip comes through by email now. So the day talk, which is really interesting, is that topics of conversation are largely practical, economic issues and planning, an enormous amount of complaint and criticism as people get on to each other's case, gossip. And people say, well, gossip maintains social norms. But in fact, I found 30 to 40% of the gossip was just total bullshit. It was constructed for social strategies. And so you can see that. That was the day. And then there's a switch to the night. All day long as an anthropologist, people are onto my case to give me things, to give them things, to help with things. At the night, that's off limits. Everything mellows off, and I don't get a single demand. Isaac Dinesen, or Karen Blixen, wrote, "At times I believe that my feet have been set upon a road which I shall go on following, and that slowly the center of gravity of my being will shift over from the world of the day, from the domain of organizing and regulating universal powers, into the world of the imagination, with the coming of the dusk, with the lighting of the first star and the first candle." And that's the transition you feel. So I looked at nights of 36 nights in 4 camps. 8 nights were singing, music, singing, gathering together. 6 nights were trans-healing dances, and most nights were conversation and storytelling. And that's where I recorded the conversations. And the results were quite astounding. All you really need to do is look at the orange here. Actually, the print is pretty big. But you can see in the day there are a few stories, economic complaint. At night, people get off each other's cases, and they mostly sit around and tell stories and come together. And so the mood's mellow when the sun's set, and I can tell you it's very harsh in the day because it's a sharing economy, and people are always mad that he gave that to her and not to me. And people gather around single fires to talk and to story. They talk about their own experiences or those of others in a very entrancing, compelling rhythmic language. The listeners are stunned with suspense, rolling with laughter, close to tears, and emotions are synchronized because everybody is relating to the same story. And the people they talk about are often people who are 200 kilometers away. And they bring that person right to the hearth. So delicious are the details of the story. But the storytelling, what I'm going to suggest here, is that it gives the big picture of society. Good storytellers transmit visions of social institutions that are imaginary, like kinship system, exchange systems. And they're not present to the senses. They're never wholly perceived in reality. But the people, the few people like this old lady, who can put stories together to imagine the big picture of society and tell about it, they become very influential. These are the organizers and the mediators who have this skill. And this is an example of Harrow exchange. This is an exchange of gifts where people choose partners up to 200 kilometers away. That longest line there is 200 kilometers. And this is a social security system, so they keep exchanging gifts, and the underlying relationship is when you're in trouble, I'll help you. You can come live with me. And when I'm in trouble, you can come live with me. So that is the exchange. The average person has about 15 or 16 of these partners. And they usually spend about 2 to 3 months a year living with these partners. This old lady is wearing all her gifts to advertise herself because she's trying to marry off her grandson. And she's showing how well connected she is. And then there are stories and give the big picture of regional gatherings, Harrow exchange, and kin relations. And the one story I collected, it was from the 1940s, 6 bands coming together at a particularly good time of year to harvest marima beans. And this story goes on for pages, and the relations between the bands are described. And the complex kinship relations are described. Everybody named would tell how they are related to so-and-so and so-and-so and so-and-so. And this gives you an idea of the area from which people came for this gathering. The whole thing would be about 200-300 kilometers north-south. And this is sort of an excerpt from the story. Different people were there. Some were there from the now Isa Valley, and some narrow from the further south. Some from the Ngwasi, where there is a forest with lots of cambridum trees. The northern people were there also. They were called Kwesi, also came. The owners of Ngwa also came. And then they tell how everyone is related. The story. And this is a story put together from many that she had heard, because she was a little girl when this happened. And then they describe what happened at the gathering. This goes on for pages. She got the other skin apron and gave it to my late grandmother, Ngakanga. She took out another skin apron and gave it to her aunt, the late Nisanga. And then she gave out more gifts. And then it was the beginning of gift giving. He, Kashaina, tried to pull the beaded headband on his head, but it didn't fit. So he took it off his head and gave it to my granny, Ngakanga, and gave other things to her. And this goes on and on and on. And you don't only have stories, but for the other form of arts, you have gifts. So that these partnerships of mutual obligation are marked by gift exchange, beautifully made, artistically made gifts. And these gifts represent relationships, but they also carry stories of who gave them to whom. They go through long chains of giving. So, they create these huge networks, so each gift would have a history in stories. And then another example would be in marriage. You don't see many marriage. When you live in a small band of 25 people, you may see a few in your lifetime, and they're infrequent, but the procedures are elaborate, they're arranged. And usually the girl is married around 13 and rejects the man and makes a big fuss. And these stories are hilarious, of old ladies telling about how they rejected their husband and ran away and destroyed the wedding gifts. And this prepares young people for marriage, knowing that the first years are going to be kind of rough. And that they'll get used to them. And then parents also learn, of all the case studies of marriage told in stories, and they know how to manage them. Okay, the other thing, which is people, Jungwasi do trance healing, in which the women sit and clap all night and sing, And the men, the trans healers, travel to the spirit world where they negotiate with the spirit, asking them not to take people away, to leave them on earth, that they love them, giving everyone preventive medicine. And in these stories, the trans healers at night will tell about how far they traveled. I mean, I was once telling people about we're sending men to the moon, and there's an old guy who looked at me and said, "Been there." So this is, you can imagine, so the transers then, in telling the stories, connect people with a shared cosmology, a shared sense of the spirit world, a shared sense of what causes misfortune. So sort of to wrap up, then day, night, and imagination. So the day you have economics, technology, nitty-gritty of social relations. The night, the stories are about imagined rules, communities that are not tangible or physically coherent in time and space. So they're imagining society in their social institutions. They also imagine, as change comes, they can also imagine, they can innovate and imagine changes as well. And then stories stimulate imaginations to construct the big picture of social institutions and relations. And very, very importantly, because we have theory of mind, because we can read what other people are thinking, they really make people imagine the thoughts and the feelings of others, so that you can really understand that people feel differently about things. And it brings a lot of understanding into it, and people put themselves in other people's shoes in the stories because they're so emotionally laden. Laden is incredible. So essentially what I found in this study is that the evening hours provide a space for the arts and for the imagination. And the arts would include also music, dance, and stories. And does this happen beyond the Kalahari? We moved up to the Arctic now. Yes, these kinds of stories are told in all hunter-gatherer societies at night. If you look through the human relation area files, there are 38 mentions of sanctioning gossip. None of them is at night. They're all in the day. For 60 hunter-gatherer societies, information on stories, songs, healing, and ceremonies is in the files. And of course, as we all know, we do hunt and gather stories through theater, film, recording, literature to feed our imaginations. I mean, we really are as addicted as anybody. So to end then, I just want to ask this question. I often think about that. What is in our society the impact of checking the phone, flipping off the light switch, with no time for the imagination to explore, to ponder, and to position events and relationships of the day in the past, present, and future? And these are thanks to my coworkers.
Good afternoon. My name is Pascal Gagneux, CARTA Associate Director. When Alysson and Sheldon and I discussed the possibility of this symposium, I immediately mentioned the Kennis brothers to them. Adrie and Alfons Kennis are world-famous paleo artists from Holland. And I contacted them and they kindly agreed to come. And we learned 2 weeks ago that due to a family tragedy, they couldn't. But luckily, we could convince them to answer about 15 questions for us in a brief video, in a filmed interview, interspersed with some documentary footage that has never been shown before. So I hope you will enjoy the upcoming presentation. Adrie and Alfons Kennis telling us how they go about making such believable recreations.
We are Adrie and Alfons Kennis, and we make reconstructions of humans. And we started because we were very bad at school. We were not good in mathematics, and we were not good in reading. So the only thing we could do was making drawings of faces and about animals and everything. And the first, I show you two examples of the first reconstructions we made. There are those two. On the left for you, on the right, you see clearly Cro-Magnon. This is Cro-Magnon man, and on the left side for you, you see clearly a Peking man. And especially when we came in the library, there were not so many books about human ancestors, but one illustrator stick out, and there was this illustrator Zdeněk Burian from Czechia. And he made this beautiful, we thought he made this beautiful, rough reconstructions of mostly, for example, Neanderthals. And we were fascinated by it because we liked the characters, we liked the rough style. We went on vacation with my parents, we went on vacation to Germany and to Italy, and we traveled through these landscapes Burian often depicted. Because Adrie and I weren't so fascinated by dinosaurs, because dinosaurs was much too alien, much too far away. We couldn't
Reptiles have no character.
And we could not imagine a world with only reptiles. We could imagine a world with humans in it, and then with strange beasts in it, like woolly rhinos, cave bears, even cave lions. And we were fascinated by this world where people lived, primitive people walked around, and they could encounter big lions, big mammoths. How do you make reconstructions? First, you need a skull. We often get casts of skulls, if you made casts ourselves, or if you want to reconstruct the total body, we get bags of all elements of mostly pieces of human skeletons, and these pieces we try to put together to reconstruct in a complete skeleton. And for example, here you got a skull and it's missing its face, and we got only a couple of bits of the face. So we look in our big spare part collection behind me. We have an enormous collection here on skulls. In the drawers are also postcranial stuff and are in it. Here you can all kind of bones and pelvis pieces. This is our garage. We're gonna look in our collection. What type of human is this? How old is it? You have to know, of course. And what type is it? And then we look in our collection if we find other individuals from the same time frame. And then we're gonna look at it. What our bits have the same type of morphology. And then we try to recreate. More or less the face on
this
the
is
skull.
this is the end result of the reconstruction of this face. So we use this one as base. We have this part and all those loose parts, and then we create a new face. If you see carefully, you see that the maxilla here's the left maxilla of this face is put here in this part, and the nasal area we used also this some piece from Atapuerca we use here. So we create a new face.
But nowadays, last reconstructions are more made with stereolithography. They scan the bones, so it's not necessary more to do this to make casts of the originals. So we scan, the bones are scanned nowadays, and we at home we got a 3D printer, and you can print out the bones without
without have to use silicons on the originals. It's even today possible to make a reconstruction from a skull who is still in situ, embedded in calcite in the cave. This one is from Altamura, and they managed to scan the skull in the cave, embedded in calcite. And then we printed out this skull, and then we were able to make a reconstruction. So this is the result of the skull, and you see these small pecks here in the face, and those pecks resemble the skin thickness in this part of the face. So if you see here on the cheeks, you see bigger pecks, longer pecks, because there's a lot of more fat tissue and bigger muscles, especially here, and this side, here you see the big mouse eater, the mouse eater muscle for chewing. And the big temporal muscle here, and the chewing, and you see that this pec is rather long because the muscle is very thick. On the nose, the skin is very thin, and there are no muscles, not so much muscles on it, small ones. So you see the pecs, the tissue thickness is very small, it's very not so big. To guide us with the reconstruction, and especially to choose the right muscle size and how the muscles are, we look at dead corpses. We dissect dead corpses of chimps, and also we can look at institutes where they have dead bodies of people, so we can recreate the muscles from modern humans. So these muscles guide us with the reconstruction of these in between human ancestors who are a little bit ape-like in the face, probably also, especially the oldest ones, but become more and more human-like. So we have to look more at the human muscles. You must imagine, we live indoors all the time nowadays, people. These people, you must imagine people 100, 200 years ago, people who work on the land, work in the orchards and things. I know people who work on the farms. In the summer, they got tent very quick. So people who live outdoors all the time, and especially in the summer, look probably pretty dark. And that's why on Neanderthals, we make all kind of skin tests. And this one looks like a very, this skin test is much too pale for a Neanderthal. It was just a test, it was a failure. We have hundreds of failures, but this one was too pale. So we try, this is not individual, but we try to do different colors to make it darker, so it looks more like a guy who is living outdoors all the time. We built our colors inside out. So we started with the darkest pigmentation, where real people maybe also have the darkest spot, the darkest layers, and then we
superficial layers,
the superficial layers, and then we make color layer after layer, more layers. And why more layers? Because then you see nice different colors in the face. You also see with this guy here. This is the guy from Jebel Irut. This is one of the first Homo sapiens, 300,000 years ago. If you see our skin color here, it's a bit. There's a variation in color. He is more orange, he is more purple, dark, he is more blackish. So if you use more layers, it looks more realistic. In films and everything, you see this kind of stuff. These are very neat. These are the first Europeans the first Africans who come into Europe. They look very neat, look very like a six-pack and muscled. Idealistic people. Also, the woman has the clothes on the right spot. But why? If we look at, if we see human evolution, we look at anthropological pictures, and we see like this kind of stuff. This kind of this kind of. Why not like this?
Yeah, instead of this.
Instead of this. And these ethnographic photographs inspire us to see how this ancient world of prehistoric man was. This is a world very long ago, mostly beyond our imagination, because we, Adrie and I, and a lot of people in this place are grown up in the Western world with a Western civilization. But this guy next to it looks, for us, a bit nearly alien for us to compare with this European guy. Strange, but for him, in his world, he's normal, and this is abnormal. Yeah. So we try, if we try to make a reconstruction, we try to gonna dive in this world. If we were maybe in a Western museum, if we think Western, you see his testicles are hanging under the shell. But if we put him in a normal museum, we put the shell over his testicles for sure. But this guy doesn't feel naked. This guy feels himself normal. He doesn't feel naked. He stands like in his world. Yeah. In his world, it is a nice piece of necklace here on his belly. Museum directors can have big problems with it to confront people who come to visit with children and families. This naked guy can have some problems. And our culture was 100 years, not 100 years ago. It's also a bit weird because I don't. If you walk on the streets this way nowadays, I think people would.
It's not that different.
It's not that different.
That this guy.
Yeah. We also do kind of contrast in it. If you see this nice picture of a nice woman, nice child is hanging on the breast, is a nice female body, but suddenly if you see it under her face, she is smoking a cigarette, and she's a bit it looks more like a male who's kind of cigarette. So you get two things in one view. You get a female like underside in this picture, and you get a male of robust. Yeah, but it makes a nice contrast, and that makes it authentic. Smoking. When we have to reconstruct a robust guy, and we see also robust guys in ethnographic photographs, then they don't stand robust, they don't feel robust, they don't stand as a hulk. So this Neanderthal we made the reconstruction. You see it's very robust, enormous broad pelvis, incredible broad pelvis, short legs. But he stands very relaxed, he stands not as a hulk, I'm a strong man. He can be shy, he can be a shy guy, he can be a shy Neanderthal.
Introvert.
And this, we also make sure that he makes contacts. It was also a bit nice to do, that the reconstruction make contact with the visitor. So our reconstruction always look at you. So you get a really the feeling you engage an ancestor, you engage somebody. Yeah, here we see an Homo sapiens, the earliest Homo sapiens. This Homo sapiens for 300,000 years old, it's from Jebel Irut. It has a rough haircut, it has a rough face, it has a strong body, but it's rather thin, but a strong body. So what we thought, we saw pictures of females who are making themselves pretty. And we thought, why could this guy not make himself pretty?
Put some stripes on his body.
White stripes on his body. So he's doing kind of maybe a female thing, of what we think maybe a female thing. But his head is very robust, so you have again nice contrast. This is the way how people stand and behave and stand if they have no pockets. You leave your hands somewhere, and even very primitive old human ancestors walked erect, had their hands free, but if they just staring at something, relaxed pose, they leave their hands somewhere, and it's very interesting on ethnographic photographs to see where people leave their hands if they have no pockets. They do not an active pose like hunting or like making.
Try to avoid it. Yeah.
Try to avoid it, of making tools, because then they have no connection with hominid, no connection with the public, and you don't see the anatomy very well. We make a lot of reconstructions reconstruction. So for each time, for us, the face has to be interesting. So what we normally like to do is building a contrast in the face, and for often kind of tension in the face. And for example, here, this child, this Neanderthal child, has a rather big mouth. She's a little bit laughing at you. She's a bit laughing, but it's not a complete easy laugh. It's not complete easy to see. It's still hiding in the face where the laugh will burst out. But she's still in the face before. She's not laughing right out. The same have we done with this face. This boy, there's a boy Neanderthal, a bit older. But he's not laughing right out. It's just there's something starts to happen in his face. You can see here 2 examples, two sketches. They look very similar. There's a very nice face, a bit woman, but there's also a nice contrast in the face. You see here, for us, we think a rather aggressive nose, with the nostrils turned upwards. Then we put some friendly eyes above it, and we give her a friendly smile. So we get a nice contrast with a bit aggressive like nose and a friendly smile. And sometimes we go too far, we can go too far. This is another sketch, and here she puts the mouth a bit already open. But we think then, when we make this thing, that is too much, because the mouth is open. You're going to look now also at the nose and at the mouth, but the mouth attracts too much attention from the nose. So that's why we prefer to do the mouth closed. So it's you see nice contrast of the aggressive nose and the friendly face.
I'll show the work that we do, and in a way it's quite similar to the Kennis brothers. We try to reconstruct parts of the Neanderthal, but instead of body parts, what we try to do is to reconstruct their minds. And one day I imagined that it would be possible, and I don't have a brother to challenge me to keep going, but I have a whole team. And this whole team helped me to achieve that, and that's what I'm going to share with you guys. So we start with the idea that perhaps we should study human imaginations by comparing to other humans, perhaps humans that have a different kind of imagination, either worse or better, just different. And we thought about the Neanderthals. And part of the reason is because we know more about the Neanderthals than the other extinct hominins. And we also share our world with them for a long time. And becomes clear, and I think you guys all learn about that, that during the time period that the Neanderthals stay on Earth, I mean, they developed these stone tools and definitely make it better, but the technology didn't go as far as that. And in contrast, we modern humans, in a very short period of time, we could create not only stone tools, but a whole set of new technology, art, and sophisticated ways to communicate to each other. So which points out that perhaps our brains are somehow different from them. So how do we study the Neanderthals and how we can start tracking these questions? So we have bones, we have endocasts that we can predict how their brains would have a different shape or volume. We have DNA, we can extract DNA from bones of Neanderthals, and we have impressions, and someone might call that art, others might not think that this is art, but they definitely left some impressions for us to try to understand. So from the endocast, this is the work from a group from Leipzig in Germany. They have confirmed that their brains, at least in terms of volume, is not so different than us. They actually have a little bit larger brain compared to what we have. But I mean, the endocast doesn't offer too much in terms of how that brain actually functions. And Sven Pabo, also from Leipzig, is a pioneer on isolating the DNA from the Neanderthals and sequence them so we can actually compare the Neanderthal genome to the modern humans. So this is all nice, but actually how do we understand the Neanderthal brain or the Neanderthal mind in a dish? So I come from the stem cell background, and in stem cells we have something that's called brain organoid, also called a mini brain, because we can recreate those using human pluripotent stem cells. And they don't look like that. They're actually a simplified, miniaturized version of the human brain. And we have a recipe that we develop in the lab that is quite good to make functional mini brains or brain organoids. And it always starts from single cells. These are similar to embryonic stem cells or pluripotent stem cells that we can reprogram from people. I can do it from my cells, from your cells as well. So from these single cells, we grow them in different media, changing the growth factors, specific growth factors at different stages, until we can mature them, and these networks would just form and become more and more sophisticated. Another thing is that, I mean, they can grow up to a ½ centimeter, and we can keep them alive for up to 2 years. So these are quite robust protocols, because not only you create a single one, but you can create thousands of brain organoids in the same batch. So that allows you to do some experiments with them. And if you cut one of those spheres, what you can see is that they maintain or they mimic some of the embryonic brain structures in humans by having a ventricular zone in the center that sends cells that migrate out and form what we call the cortical plate. As these organoids age, these cortical layers will become more and more well-defined. So this is a snapshot of all the cell types that we have if we just age these brain organoids for about 4 months. We have mostly glutamatergic. These are excitatory neurons, and you start to see 2 populations in there. So these would be upper and lower cortical layers, but we also have inhibitory neurons as well, a small population of GABAergic neurons. There are progenitor cells that continue to produce, and at that stage we start to see some glia cells or astrocytes. These are the cell types that helps on the formation of these complex networks that I'm gonna show you how they behave later on. So this is all good, but I need live cells to recreate those brain organoids in a dish, and the problem is there is no live cells from the Neanderthals. So that poses a big problem. How can we recreate the Neanderthal mind if we don't have the raw material for that? So we turn into genetics. Since we have their genome, one thing that is possible to do is to compare the genome of the Neanderthals to modern humans, and we have thousands of them, and ask what are the differences between those genomes? What is specific to modern humans that were positive selected in us compared to them? So if we look across all the genes, we end up with about 200 genes, and this is many people have been mining this data set, and these are the genes that are different between modern humans and Neanderthals. And instead of looking for all these genes, we decided to narrow down this list to 3 genes, because we asked, what are the ones that are highly expressed during early stages of neurodevelopment, and what are the ones that we know that is, if they are mutated in modern humans, will lead to a neurological disorder. And these are the 3 genes. These 2 here are synaptic genes. They will help how the synaptic or the connections between neurons are formed. And that one is a very interesting one, because it starts very early, and it's a master regulator of several downstream genes. It's a splicing regulator, a splicing protein. So the way we do that is using genome editing, and we use CRISPRs, where we can go back to one of our modern human cell lines, and we can change or swap the genetic information from the modern humans to the Neanderthal. So we Neanderthalize the cell lines targeting those specific genes, and at the end of the day we have Neanderthal brain organoids, or Neanderthoids, how we call them, and we can compare. So, I mean, done that, we recreate that. And I'll show you some of the differences that we observe by comparing them with the modern humans. So the first big difference is at the gene expression level. What are the genes that are up or down regulated compared to us? And we have a couple of them. The interesting part is that they are all connected to brain development in several pathways that leads to brain formation. And I'll point to some of them here. This is just a list of interesting genes that we are trying to understand. There are genes such as nesting, which is a progenitor cell, early stages of neurobrain formations. There are genes such as TBR1. This is a gene that has been implicated in autism, for example, and they are downregulated in Neanderthals. And there are genes like OTX that, when upregulated, might prone you to have seizures or epilepsy. So this all happens without genetic modification. I mean, it's just by changing those early genes that you affect downstream genetic signature that might lead to problems such as autism or epilepsy. So I told you about one of the genes are actually a master regulator and controls how gene expression or splicing is regulated. And this is a gene called NOVA1. And this is a reconstruction of the protein attached to the RNA, and exactly the base pair from the DNA that changes altered that conformation, and as a consequence, the RNA splicing would be affected as well. And we are looking into these alter splicing factors, and we realize that there is a lot of information in there. Some genes like HOMER1, this is another synaptic gene, have different isoforms between humans and Neanderthals during development. Other genes, and these are actual target of NOVA1, such as GNAS or PIN1, so these are genes involved in epigenetic regulation, for example. They create a new isoform that's only present in Neanderthals. Modern humans lost that isoform from that specific protein. So these are quite interesting observations at the gene expression level. What happens to these brain organoids if they have that? So the morphology is quite interesting. They start quite similar, but as they progress in time, you can tell that the morphology changes a lot. So while the modern humans' brain organoids become a sphere, increase in size, and start to mature, the Neanderthal brain organoids have this kind of popcorn shape. And you can measure the diameter of them, I mean, how they grow, and already we can see that the Neanderthal organoids probably have some kind of cell death or some problems to develop compared to modern humans. Also, the size of the sphere is different, but also, I mean, the way those spheres are shaped, which might point it out to some differences, as I said, in cell death or apoptosis, but also in migration, the way the cells migrate. So just by looking at these pictures, we can tell that there is migration defects, and there is a way to test that. You can plate some of these cells and let them migrate by themselves. And what we observe is that the Neanderthal cells, they have a defect in migration. They migrate is lower, but they go farther than human cells. So even a small migration might affect your entire brain trajectory during development. So if you look at the neurons, I mean, we also see differences in specific subtype of populations from the cortex. This is one of the cortical layers, and we observe that SATB2, it's a neuron that participates on intracortical communication as well as intra-communications between layers. It is dramatically reduced in Neanderthal cell lines compared to controls. So everything is all about morphology and how the cells behave or the shape of these organoids. What about the function? Can we actually measure how the communication or the network is being affected? So to do that, we use a tool that is called a multi-electrode array, where we plate these brain organoids into a multi-electrode in the bottom of those dishes, and we can record. We gain an activity map as well as a raster plot. In every trace here, it's when a neuron spikes. And if you have several spikes at the same time, we call that a burst. And when bursts spikes in the same channel we call a synchrony event. And since we can keep them alive, we can see that they evolve. And we did that. And when we start, we actually didn't have what to compare. So I'm showing here just some of the work that we did in 2D. This is without the three-dimensional structure. And most of the data, it is below 5 Hertz. So this is just an average measure of the brain activity. So what is the idea? What is our goal? So a mouse brain is about 18 Hertz, and a monkey brain is about 20 Hertz. So other people have also created different protocols for brain organoids, and although they can also keep them alive for a long period of time, they were not able to see an increase in activity, suggesting that there is some problems with maturation. But we are glad that the protocol we have, we were able not only to keep them alive, but to reach a level of activity that's much higher than was ever recorded before in vitro. So if you have that much level of activity and synaptogenesis going on, it means that you can record something else. And this is what we call brain oscillations or brain waves, the activity that you can actually measure through the skulls using electroencephalogram or EEG. So we can actually measure those EEG waves from these brain organoids, and we can compare them to preterm babies. So we created this machine learning algorithm, and we trained that in an unsupervised way. By exposing them to different EEGs from preterm babies and asking the machine to tell the age of the person. So once the machine has done that, we feed them with our brain organoids EEG, and we ask what are the predicted age of the organoids. So this is how the machine will do in a perfect condition in the red trace. The data is actually on the black trace as well. You can see that we have a very good prediction up to above 25 weeks. And the reason why the machine is not good to predict below that is because we simply don't have any data from human preterm babies below that. The babies will die, and we don't have a way to record in uterus that is non-invasive. So that's why we don't have a good prediction. But what is the prediction that we can have? And I'll show you just one example of one of the parameters from the EEG, which is the space between the interval of activities. So if you just look at an adult EEG here, so it's quite complex. This is a preterm baby. You see this spontaneous activity transient followed by a quiescent period where the brain is just silent when you're super young. And here is in our brain organoid. So the brain organoid is much similar to a preterm baby than to adult baby. So that's what we are modeling. We're modeling early stages of neurodevelopment. So as we grow older, the interval between those activity becomes shorter and shorter. That's why we have this complex EEG in adult. So the organoid, as we age the organoid, the interval also reduces to a level that gets close to that red bar, about 40 weeks on 9 months. That's equivalent to a newborn, healthy human baby. So that's what we can get with these human organoids. So now that we have lots of activity to compare, I mean, how does the Neanderthal brain organoids compare to modern humans? So we have been studying that from different angles, and I'll first start by showing what is the level of synaptogenesis, how many brain cell connections the Neanderthals can make compared to modern humans. And we see that there is almost 50% reduction, which is similar to someone with autism, for example. So definitely a lower level of synapses compared to controls. We also measured them on the multi-electrode arrays. And what we have here in this part is just the raster plot, and the activity map, and then some of the quantifications that we have. The number of spikes, the level of activity per neuron per minute is way reduced to what we expect for an isogenic control. The same is true for the firing rate for these neurons is also reduced compared to isogenic controls, which again points to the fact that these networks are just not good enough or not mature enough to the level that we would expect for modern humans. So I I hope I could tell you that by recreating this Neanderthal brain in a dish, we could say that they have differences in morphology, synaptogenesis, even at the network level. But the question is, are those differences better or worse than the modern humans? And I made like some analogies with autism and other neurological disorders, but the truth is that we don't know. So we have been looking at using different experimental models, and I'll just show you one thing that we are excited about. And this came from this idea that if you have brain oscillations from people working together, they might actually synchronize, or they are more prone to work if your friend is with you than a stranger, which suggests that to achieve something, to achieve a specific goal, it's better to work with someone that you like. So we have been tagging these human brain organoids into robots. So that's a way for the robot to experiment the world. And we are doing that in two ways. The first step is just teaching the organoid how to move a robot. And that's what you're going to be seeing in this video. So this is a robot that has been moving all the legs just by the frequency of oscillations coming from a human brain organoid. So the idea is now to do the second step, which is to create a feedback loop so the organoid can actually learn something and the robot can tell by experiencing the world. So soon we're going to be doing the same experiments using Neanderthal brain organoids, so we can see if the robot can actually learn something better or not than humans. So I'll finish here by summarizing that we have a model, a brain organoid model coming from stem cells that can recapitulate human development. We can reconstruct evolutionary steps that recapitulate key features of the Neanderthal development. We think so. And this might be a paradigm for learning if you put them back to a robot or something. So these are Kleber and Priscilla. These are the brain masters in the lab. They are the ones who developed this nice protocol that everybody's excited about. And these are my collaborators for this project, both Katerina Semendeferi, your lab, the Wojtek lab, and Eric Green, who help us on the genomic analysis of the Neanderthals. And these are my funding agencies, and I'll stop here. Thank you very much.